Welcome, everyone. It is a great pleasure for us to have you here today. My name is Adrian, and here's my colleague Julia, and we'll be your host today. If you're new to this platform, please take a few seconds to locate the Q&A and chat boxes. I also want to remind you that this session will be recorded, and feel free to submit questions anytime throughout the presentation, and we'll be reviewing those questions towards the end. Yeah, let's get started. Before we dive into the topic, I really want to introduce you first to Cytena. I think it will be especially helpful for those who are joining for the first time. Cytena's expertise are focused on cell line development. We have more than 200 customers globally. They trust our products, and we offer a range of benchtop instruments, consumables, and reagents, mostly around cell line development and related fields. We were founded in 2014 as a spin-off of the University of Freiburg, starting with a unique technology of single-cell dispensing. And in the past 10 years, the features of these products, especially around the core technology of single-cell dispensing, has been improved, and the portfolio has been expanded with complementary products and reagents. And basically, our goal is to make laboratory processes more efficient and bring in therapies to patients faster, safer, and more affordable. Okay, having said that, I think it would be nice to know what we'll be talking about today. And the way would be, we'll be dividing this presentation into four major parts. And I have to say that most of this is driven by insights from our application team. They have generated this knowledge and two customer insights as well. The first part, we'll talk about the two most common cell lines, and followed by how we could or how you could optimize those the cloning of those cell lines. Third, we will show you examples of how customers are working on this relevant field. And finally, we will wrap it up with Cytena's latest developments. So basically, a lot of it starts with the selection of cell lines, and that's basically how everything will get started, and knowing how to work with this alliance is very important to optimize your processes. This brings me to our Cytena's core focus, especially because if we're going to be talking about cell line developments, I want to highlight two of these focuses, and that's working with monoclonal antibody production and other therapeutic proteins. The second one would be viral vector production that are used typically for gene and cell therapy. Of course, Cytena also is able to work with stem cells, such as APSCs, but this would not be today's topic. What we do is that we support all the areas around the knowledge and expertise of the single-cell cloning part, and this experience helps us to provide you with in-depth application support and protocols. And specifically for today, we'll be focusing, as I said, on these two first applications, but please stay tuned for upcoming events from Cytena, especially on the content of the know-how to do things, right? Just so we are all on the same page, just want to mention the overarching goal of the cell line development, right? Of generating a stable cell line development, starting with genetically engineered cell lines in order to produce biotherapeutics such as antibodies, viral vectors, and so on. You start from these genes, genetic cell lines, until you get into the banking of cell lines, right? And this whole process typically takes about 6 to 8 months, depending on the level of automation. And of course, there are plenty of very comprehensive and stringent requirements around monoclonality and all that you would need also to create content or reports for documenting your process. Another big part that we haven't really talked about too much is the media, protocol optimization, culture conditions, right? And that's what we are going to try to focus today. All right, to give you a more precise overview of exactly what part of the cell line development we're talking about, I'm just going to show you these two general workflows. You may be familiar with the top one, where you're using limited dilution and you take multiple instruments to do you image what you have seeded in the plates, another instrument for cell counting, another instrument for titer. And here today we focus on an instrument called the Up.Sight second generation that can basically help you shorten these timelines, and also instead of using 3 instruments or more, you can carry a lot of the steps in one device, right? We will not go today into the very specific or the specifications of the Up.Sight, but we have already some content, webinars that you can also look into for Cytena. But rather I want to focus on the actual cell culture aspect of it and efficiency of cloning. Let's start from the basics, the 2 main cell lines that we will talk about, and that's CHO cells and HEK cell lines. Basically, how do you select those cell lines? They're mostly driven by the primary use, right? As an example, you'll be looking for cell lines if you're HEK cell lines if you're going to work under viral vector production or gene therapies. Of course, CHO cells have been extremely useful historically, right? With a lot of regulatory approvals already backing them up, right? And yeah, another comment, if you're using another cell line in this field, I would be very happy to hear about. Please type it in the chat. We'd just be interested also to know what else we can support and also what kind of content we can create for you in the future. A lot of our customers are working with us particularly to ensure high clonal recovery, and this is often related a lot to the survival rate during the single cell dispensing. This is a very critical step for this. In this case, both cells can be very fragile and prompt to dying during the dispensing, the single cell dispensing. This could be because of the shear stress that the cells experience during this step. And I would like to argue that the Up.Sight is one instrument that definitely helps here. But of course, there are other things to consider. These 2 cell lines, of course, are aimed to be grown in suspension and need to have low shear stress during the culture process as well. And there is a strong need for culture optimization. And although not a relevant process to the sorting process, scientists also look into genetic stability and the long-term impact of such cell lines. And finally, scientists and engineers, bioprocess engineers, are aiming for identifications of clones with high producing yields, right? So you want to optimize for that as well. And that often comes back to the optimal growth conditions, right? Having said that, I would like to highlight the relevance also in what I just said about clonality, and that's because clonality can also affect directly and indirectly some of the optimization steps, and this is why there are such requirements, right? Clonality supports product quality, safety, drug efficacy, and in addition to being a part of the regulatory requirement. Finally, also extremely important, all of these impacts also how the cost effectiveness of the whole process, especially when not done correctly. This can really minimize or increase costs. For take-home message, scientists are seeking for reliable processes and technologies. So I think we can agree that cell culture and cloning optimization is cell line development also. So that's how we want to look into it. And clonality plays a big role into these multiple steps. So let's go over how also Cytena can help. And I would say this is also 2 cases that I would try to bring some value to you. So now in part 2, we want to provide you with tips on how to optimize cell recovery. Before I go into the specific details, I want to tell you that all the tests have been described here were carried out with cells 293F. These are from Temor Fisher. We believe this is a good example of a typical HEK 293 derived suspension cell line that is often used by our customers. But some of these recommendations could also be extended to similar cell lines. I also want to make sure that you understand you could do more exhaustive testing in addition to what I will show you. So we first start by thawing the cells, the suspension cells in pre-warm freestyle 293. They were transferred into a media of 20 milliliters into a shaking flask. Then we allowed for at least, I would say, two days for cell recovery, and always checking that they maintain high viability before splitting, of course. Splitting the cells when they actually reach up to 3 million, ideally 2 million cells per milliliter. And one interesting finding was to not let the cells dilute below 200,000 cells. And for the cloning step, we tested the freestyle 293 expression cloning medium, and this was in the plates that we filled the plates with 50 microliters per, which were also pre-warm. Then here we have a comparison of the results, I would say with and without cloning media. I think that's especially very strong when you see that the conditions are very obvious, I would say, and the benefits are pretty clear. To say yes. Second part, we also look into how to prepare this cloning media, right? Because I didn't show you how this was also optimized, another process. I know it's not always possible to have conditioned media, but definitely if you have the chance, this would be a way. We looked into what would be the best cell density conditions, and then we found an optimal condition around 1.5 million cells as the best seeding condition for this protocol. Again, you could optimize also for potentially for different components, but in this case, we looked into conditioned media. Another key aspect of success, I would say, was not to disturb the cells in the first few days after dispensing into the plates. And so basically just leave your plates in incubator, don't touch them for about 7 days. That increase also viability, recovery, cell recovery. And one final step was looking into the well plates, whether they treated versus not treated, what would be the effect. And we've taken this experiment basically comparing two treated plates and two non-treated. So you see these labels also, the exact part number of the plates we have chosen. Something obviously interesting was how the colonies, the morphology of the colonies, and we can also look at the non-treated ones, how this clumping behavior of the cells when dispensing on these non-treated plates. But it's surprising you can still also derive viable and robust clones even from those clumped colonies and scale them in shaking culture conditions. So of course, even if you have not taken the correct plate or the most optimal plate, you could still grow them. Now I've come to the end of the HEK saline part, and I want to show you a second example, but for CHO cells. So this is now the work done for the CHO cells. Also, if you're interested in the full paper, I have written here at the top the publication. You can find it in the bioarchive. Here we have followed somewhat of a similar process, but adapted to the cell lines. We started also again with highly viable cells, and we have explored the impact of cell culture in cloning media. The cloning media, in this case, lean, show the better clone recovery and more homogeneous recovery as well. And then the second part was to study the impact of 3 different cloning media. And you have these combinations here in the graph plotted, and CHO led to the better clone recovery, I would say, than the other 2 tested. So basically here, the data is suggesting that a careful selection of cultivation and cloning media can improve your clone recovery. One typical supplementation strategy, as we saw before, was using conditioned media. For example, could be fresh medium from medium used to grow other cells. That's what I mean. And then, or this media contains different growth factors, signaling molecules already that can improve basically the outgrowth survival rate, right? In this case, when we looked at a case of 80 to 20% ratio of conditioned media to fresh culture media, we saw an improvement. The performance, of course, vary depending on also on the raw and conditioned media. the media used behind, right? So from 10% up to 60% variance, yes. What we did as well was to take this similar protocol and just try it on different cell lines, right? And we were able to optimize conditions, cell culture conditions for several suspension cell lines. In this case, they were all CHO derived cell lines. You can see here they were all CHO express, CHO K1, CHO S, and of course we believe this would be the way to optimize your culture conditions. And finally, we also did a similar test as I showed for the HEK lines, and we were looking into defect of the well plates, and clone recovery is obviously affected by the surface of the well plates. And as you can see, the results are variable on this case. It's not only the surface of the well plates, but also other aspects such as the shape of the wells and things like that. And that's because you also want to think about the influence of evaporation rates in general, right? Particularly on the outside or the outer wells can be more exposed, for example, right, to evaporation, and that would impact clone recovery and health. I think what's also interesting in this part is that we show you now some results for two types of well plates for 384 and 96. Before closing this discussion, I also want to bring back to this slide here, which is the slide I showed you before at the very beginning, how Cytena can also bring some value to you and working with us, how that really looks, right? And that means that we have some nice guidelines on how to culture cell lines and clone optimization. And we understand, of course, they are cell line dependent, right? The second part is that clonality reduces risk, right? And it needs to be documented. And that's something that Cytena excels at. We have developed a lot of tools around documenting clonality in a way that it's easy for users to implement. And having said that about clonality, I want to bring your attention further to optimizing imaging for purposes of clonality and other aspects of the instrument itself as well, not only the cell culture aspect, right? And this comes back to this first slide that I showed you. Why I mentioned the imaging is because we also checked certain aspects of the Up.Sight. Of course, these are just a few of these well plates, and we will next year potentially release something similar for a more exhaustive test. But here, this is something actually that you could also do for your particular cell line to think about some kind of analysis that it's also considering clone recovery or the instrument that you're using, right? Or even the cost. In this case, we also were looking rating plates in terms of cost benefit, right? And even quality, right? So you have to think about, yeah, effects of plate surface, evaporation rates as well. This is a limited assessment, but again, I will encourage you to look into a similar analysis at home and over your protocols. Yeah. So I hope this can serve as a good guideline of what steps you could do to optimize your workflow. All right, now let's go over how we are using the Up.Sight in this process. And I will do it just first by highlighting a few aspects of the Up.Sight, and then we'll have a look at the two customer insights. So what can you do with the Up.Sight, right? So all these optimizations that I show you can be performed with the Up.Sight, and we would utilize the capability of single cell dispensing and a very efficient single cell dispensing, resulting in easy clonal derivation. It's a gentle dispensing, right? So you would increase also recovery. And a good interesting point again is that you have with this new instrument other features such as wall bottom imaging that will help you look into confluency, cell count, and this instrument also works with multiple tools that allow you to measure titer for CHO cells, for example, that are producing proteins in monoclonal antibodies. And it works with with an instrument with a software that will support the documentation for clonal derivation. Although I'm not going into very in-depth details of the Up.Sight, I will just highlight a few and show you how it does the clonal assurance. So this is all possible. First, the dispensing is done or analyzed basically via nozzle imaging. You have your cells in suspension and you have a camera looking at the nozzle and look at what is dispensing. The second part could be looking into the well plate and imaging the cell as it's sinking through the plate. That's what we call the 3D well imaging. Or alternative, you can also use the capabilities of the well bottom imaging of the instrument. And in that case, you are letting the cell sediment to the bottom of the well plate, and then you can just image a single cell. And that's how it's working to create this double assurance of clonality. Again, I'm not going into much more specifics of the instrument, but rather I want to show you or tell you 2 stories of our customers working with the Up.Sight and other products, other single cell dispensers from Cytena that are working on this specific field that we just talked about. The first would be a company called Vive Biotech. They have actually developed their PCL with the Up.Sight. And as you can see, they've taken multiple steps for that. And they basically have used the Up.Sight for the subcloning of their cell lines. And they were after screened for functionality and genetic profiling. I will not show you all this data, but definitely I have here the reference. You please have a look. It's at the very bottom of the presentation. And again, the whole process was to eventually generate a stable cell line. I will show you a second part from this paper, and that's basically how they've used the Up.Sight also to generate the clones and then screen plenty of clones in order to pick the best clones, again based on LVV productivity, genetic profiling, and so on, not just based on imaging, right? Capabilities. But here they have used well plates, 296 well plate type, and they have used also our software C-Studio to generate all these images and collect them into an assurance of clonality report. The second case is New Biologics and their, I would say, multi-imaging strategy to directly generate two cell lines. In this example, here they have used the Up.Sight 3 ways of imaging. Before dispensing, it took a picture before dispensing. Here you have the images of the nozzle during dispensing, and they have used the 3D stack imaging for that. And I would say this is also a very nice story of success. And you can see here how the cells would also look like under 3D imaging. Feel free to check out their website as well for more information. Now I want to come to the future directions or current innovations as well that are ongoing at Cytena. And on you, I would say the data generated this year, but a lot more is coming. And one big aspect is the upgraded well bottom imaging of the second generation. And this doesn't matter if you're working with suspension or adherent cell lines. You can see here a few examples. Here is suspension, adherent cell lines, looking very sharp, and you can trace colonies very easily with the Up.Sight. A second part is also the extension of being able to image different well plate types or different size, I would say, right? So here I give you a few images for you to look at, starting from 384 down to 6 well plate types. Again, doesn't matter which plate format you want to image. Here it would take about 6 minutes. Another great aspect of the Up.Sight second generation, which you might have been somewhat familiar this year, is its integration into a fully automated cell line development workflow. And here you have what we call the C station, right? This is one picture of it and how it could look like for you and what it contains. We have integrated powerful software with Cytena automation suite, including C Studio, liquid handler, plate handling robots, incubators. They could be also shaking incubators. And of course, we work with Biosero for automation. Just a quick mini video that you could see a little bit of what's inside and how the robotic arm is moving plates between the shaker, the liquid handlers, and the Up.Sight. The last improvement, it's around extending the capabilities of C Studio. And C Studio has become way more powerful, consolidating a lot of data from the Up.Sight second revision and extending the tools of confluency, cell count, titer, and generating these hit pick lists that are pretty easy for clone transferring. And this basically extends clone traceability, clonality reporting, right, that are ideal for NDA and IND submissions to the FDA or EMEA, and having software that supports modified data supports CFR Part 21 Part 11, and that will help you integrate these workflows into GMP workflows, right? Now that I mentioned Confluency, I wanted to show you examples of this and what it would look like for the combination C Studio Up.Sight. And this would be one nice example of our Up.Sight reading this. I've come to the end of it, and I want to show you, summarize some of the content basically. And it's clear that optimization steps will depend on the cell line of interest, right? And even if you just talk about specific CHO cells, they will be genetically engineered, and their viability and survival rate will depend on so many things, such as the cloning media, right? And that plays, especially, I would say, significant role in clone recovery. After we have demonstrated to you that you can optimize that clonal recovery, and I think it's a nice guideline of what to look into. Can I use conditioned media? Can I supplement? Can I look into different plate types? We have done the same for the HEK lines, right? So that's a similar process that works across the board. Plate optimization, it's important to, of course, take the technical aspects, but also financial considerations. Also showed you two cases where customers are using the Up.Sight for their workflows, and I would say what is now the innovation part of Cytena is bringing a much more powerful Up.Sight, the second generation with C Studio extended, I would say called extended, because it has a lot of more capabilities to manage data at a different level. And it can now be, all of it can be integrated into the C Station using the latest technology. I want to thank you for being here and listening. Feel free to take this information, this contact info, if you need to get in touch with us for a consultation, or maybe you're a customer and you just need maybe further guidance to optimize your workflow. What I've shown you is somewhat limited, we understand, and there is more to come, and we are working hard on getting more data for you. Thank you so much.